Today we are going to see plant physiology, and hope the weather, everything is conducive for our class. We have a fantastic weather here, so I hope it is the same with in your places too. Okay, so with that we'll start our class on. Today we are going to do as you all are, some of you all in matriculation schools. So for you people also it will be very helpful. We are going to do glycolysis, TCA cycle and electron transport chain in one go. Okay, so we are going to see this in a coordinated way from the glycolysis till TCS tricarboxylic acid cycle, and then we are going to go ahead with till electron transport chain. As you all are all the more familiar in your schools, you have learnt about glycolysis, TCA cycle and electron transport chain. Let it be a revision also. Okay, so shall we start our session on this physiological aspect? Not only it is there in happening in plant cells, but it is also very, very vital for animal cells also. So we are going to see how to burn glucose. Okay, how to burn glucose to extract the maximum energy. Okay, so how to burn glucose to extract the maximum energy, to extract the maximum output in the form of ATP is the talk of today. Okay, so what are we going to do? We are going to see how to burn, how to extract, how to burn glucose so as to get maximum energy. This is what do we call this as? What shall we call this as? So how to extract the maximum energy out of glucose? What do we call precisely? What term do we use for this? Can you give me a term that we use for this? Yes, go ahead. Fill the blanks which I have given you. Fill the blanks which I have given you. What are we going to talk? Yes, we are going to talk about how to burn glucose. This is a process of this one. Okay, yes. What is it called? What is the burning of glucose to extract maximum energy can also be called as this one. What is it called? Kamraj School has wonderfully answered it as aerobic respiration. Fantastic, Kamraj. You have answered it beautifully. That is, it is none other than aerobic respiration. Well done. Yes, well done. It is aerobic respiration. What you say that how to burn glucose to maximally take in the energy is called aerobic respiration. Now we are going to see a question in that perspective. Then we will have a detailed discussion of how to burn glucose to till mitochondria. Okay, we are going to see that entire 3 processes in one go. So before that, a question in this aspect. So I am going to pose a question. This is not my own question, it is a previous year NEET question in which they had asked, when you compare anaerobic respiration with that of aerobic respiration, take the question, when anaerobic respiration is compared with that of aerobic respiration, or aerobic respiration is compared with that of anaerobic respiration, the efficiency is 2 times efficient, 18 times efficient, 32 times efficient, 36 times efficient. Okay, I repeat the question. And when you compare aerobic respiration with that of anaerobic respiration, okay, this is aerobic. Okay, aerobic respiration with that of anaerobic respiration, the efficiency of aerobic respiration over anaerobic would be twice, then 18 times, then this is 32 times, and that is 36 times. To answer this question, okay, we have to go on a journey from glycolysis to TCA cycle to electron transport chain. So we will reserve the question and then we'll come back to the question towards the end of the session, and then we will try to answer this question. Okay, so we will try to answer this question. We will see it in the end of the session. I will repeat the same question towards the end of the session. Okay. Now we are not going to answer that question. Okay, reserve the question, keep it for yourself. In the end of the session, we are going to see what could be the answer for that. Okay. What was that? I think I'm getting a response. Okay, I'm getting a response from MRS. What was the choice I gave you? I think it was 2x, 18x, then 32x, then 38x, something, 36x, right? So we will reserve this question. We will see it in the end. Okay, we'll reserve this question. Yes. Okay. So we're not going to answer. D is not the right answer. We are going to reserve the question. MRS has answered a response. Okay. So 36 times efficient. We will see the answer towards the end of the session. Okay. So now we are going to go ahead with glycolysis so that we will come back to answer this question. Okay. Got it? Yes. Now, as of now, MRS, I'll tell you the answer towards the end of the session. Okay. So let us go ahead with the process of glycolysis, which you all are very much familiar, right? You all are very much familiar with how to burn glucose, so it is easy for you also to do it simultaneously. When I write the structure, you also can start doing the structures. Okay, so start doing the structures. So we are going to start from Embden-Merov pathway. So take your pen and paper. So this session cannot be done without pen and paper. All of you take your pen and paper and start the Embden-Merov pathway. So it can't be a chalk and talk class. So you have to do it yourself. Okay. Yes, all of you are ready. I'm sure that by now you all have your pen and paper with you. Then start Embden-Merov pathway along with me. Okay. Yes. Shall we start Embden-Merov pathway? So we are going to do this Embden-Merov pathway. Okay. So this Embden-Merov pathway tells us how to burn glucose so as to get pyruvic acid. So one molecule of glucose is going to be burned, and then we are going to reap the product as 2 molecules of pyruvic acid. I reap 2 molecules of pyruvic acid. And this is a 6-carbon compound. This is a 6-carbon compound, whereas this is a 3-carbon compound. So 2 C3 carbons we are going to get from one hexose. Okay, glucose is a hexose, as you all are aware. So the hexose is going to give a triose, 2 triose molecules. Okay, that is what we are going to look at now in the EMP pathway. Okay, Embden-Merov pathway is nothing but glycolysis. Glucose is going to be chopped. Lysis of glucose. Okay, glucose is going to be lysed. Okay, that is why we call it as lysis of glucose. We call it as glycolysis. Okay, glycolysis is nothing but lysis of the glucose molecule, and hence we call it as glycolysis. Okay, so with that context, we'll now proceed with the 10 reactions of glycolysis. Are you all ready with me? Have you all started? Yes. Okay. You can even take a snapshot of your workout and show it to me, project it also. Okay, so that also can be done. So you can take a screenshot and then show it to me, and then we can verify the structures. Shall we start? Yes, glucose. We are going to start from glucose. As you are aware, glucose is the primary source, transportable form and utilizable form. Okay, in the case of animal kingdom. Okay, in animal kingdom, glucose is the best, transportable form and utilizable form, whereas if you look at other plants, the storage form is in the form of starch. Okay, and the storage form is in the form of starch, and the transportable form of glucose will be sucrose in plants. Okay, for us it is glucose. Animal kingdom it is glucose, because brain needs glucose, heart needs glucose, kidney needs glucose. Okay, the intestines, everywhere requirement of glucose is high in animal kingdom, whereas the requirement plants do require sucrose, which is quite transportable form for them, and the utilizable form is of course glucose, but storage form will be in the form of starch for them, whereas in the case of animals it will be in the glycogen. So glycogen is the storage form, utilizable and transportable form is glucose, whereas in the case of plant kingdom it is storage form is starch, utilizable form as well as the transportable form is sucrose, utilizable form is glucose. So with that context, let us go ahead talk about this first 10 steps of glycolysis. Okay, so now we are going to do glucose C6H12O6, right? So it is already, we have done the structure of glucose in biomolecules. Okay, it is an aldohexose, right? The functional group of this glucose is aldehyde. So it is an aldohexose. So one molecule of glucose gives rise to, so we are going to have, let us have the enzyme and the byproducts. Okay, in another color. So first reaction itself is an irreversible reaction, and again there are 3 rate limiting steps. Okay, there are 3 rate limiting steps. The first step, the third step, pivotal step is third step, and the last step. So the first step is catalyzed by hexokinase or glucokinase. Third step will be catalyzed by phosphofructokinase, and the last step will be catalyzed by pyruvate kinase. So all kinases belong to, okay, I'm posing a question, I'm making you recall what we saw in biomolecules, because we are going to here talk about kinases, right? So in this, ATP is going to come inside and then liberate ADP. So that high energy present in the gamma phosphate is going to be donated to glucose, so that the glucose becomes glucose 6-phosphate. The enzyme catalyzing is hexokinase or glucokinase. Hexokinase or glucokinase. Okay, specifically glucokinase, or any sugar, it can be hexokinase. Okay, so let us go for the first step. Now in the first step we said that the kinases involve transfer of phosphorus group to another one. Okay, transfer phosphorus group to the reactant so as to get a product. So the phosphorus gamma phosphate present in ATP is donated to glucose, and then glucose becomes glucose 6-phosphate. Now tell me, with your understanding about biomolecules which we did some time back, okay, so a few classes back, isn't it? Answer me, this kinases belong to, kinases can be categorized in, belong to A, B, C, D. Class 1 enzyme, class 2 enzyme, class 3 enzyme, class 4 enzyme. Yes, who can answer me this? Yes, before we go to the next step. Okay, so now, as of now, you saw that the gamma phosphate present in ATP is donated to reactant for the reactant to become product. So this kind of reaction catalyzed by, just now as I said, first reaction, third reaction, 10th reaction. They are all rate limiting steps. These 3 rate limiting steps catalyzed by glucokinase or hexokinase, phosphofructokinase, and the last one, pyruvate kinase. They are all catalyzed by, okay, a particular enzyme. Which class does it belong to? Yes, can I have the response? Which class kinases belong to? Kinases belong to? Kinases belong to which class? Yes, global, MRS, can I have the answer? Kinases belong to? All kinases belong to. Can you recall the 6 classes of enzyme I have taught you? Recall the 6 classes of enzyme, because we did only 6 classes. I said we should never change the order, right? So I told you not to change the order. Can you recall the 6 classes of enzymes? Recall the 6 classes of, who can type me and show me the 6 classes of enzymes? 6 classes. Remember, we did 6 classes. 1, 2, 3, 4, 5, 6. In that, which class the kinases belong to? That is my question. Yes? Go ahead. What are the 6 classes? Can anyone recall? Global, MRS, where are you all? Can you recall? Yes. As usual, your vibrant answers. Expecting your vibrant answers. Yes? Come on. Tell me type and show me the 6 classes of enzymes that we have seen some time back. 6 classes of enzymes. What are the 6 classes of enzymes? Yes, I'm not getting an answer. Yes, global, where are you? MRS. MRS and global. I anticipate your response. Okay, yes, first one is oxidoreductase. Fantastic. Class 1 is oxidoreductase. Very good. Then class 2 is transferase. Transferase, you have missed one. Transferase. Then after class 2, what is class 3? Hydrolase. Hydrolase. Then class 4. Class 4? Hydrolase. Yes, of course. Class 4. Then class 4? Lyase. Wonderful. Lyase. Wonderful. Then class 5. Class 5. Isomerase. Very good. Very good. Isomerase. The last of all is? Very good response. Beautiful response from whoever it is. I don't know who responded me that swiftly, the order in which we have seen the enzyme. Beautiful response from the school, I think it is KG, right? KG and MRS, very good. So this is the order, and we said that we should not change the order because this goes like this, okay? Like International Union of Biochemistry standardizes the enzyme nomenclature, and we should not change the order, as I said. So which class kinases belong to? They transfer the phosphorus group which is present in ATP, donates it to glucose to become glucose 6-phosphate. So it should be a transferase. Okay, it should be a transferase. Okay, so you have to understand it should be a transferase because it transfers the phosphorus group present in ATP to glucose to make it glucose 6-phosphate. So hence it is a transferase. Okay, it is a transferase. So class 2, the answer is class 2 enzyme. The answer is class 2 enzyme. Okay, now shall we go for the next step? So having understood, we also have to rope in all the knowledge about enzymes here. Okay, so we have to bring in all that. Okay, so the first step, as I said, is a very, very vital step in conversion of glucose to glucose 6-phosphate. Glucose require transporters to shuttle inside the cell and outside the cell. Okay, so they do require special transport mechanisms, unlike diffusion of gases, diffusion of water. Glucose required glut. Okay, glut, that is glucose transporters present in the plasma membrane so that it can be transported inside the cytoplasm. So this very event of glycolysis, again I repeat, this very event of glycolysis. Okay, so here is the plasma membrane, I said, has glut, that is glucose transporters, which will transport the glucose from outside to inside. Okay, so they are all just bilayer structures, and we have in the bilayer structure we have a membrane protein which transport the glucose from outside to inside. Okay, so glycolysis takes place in the cytoplasm. I repeat, glycolysis takes place in the cytoplasm. Okay, cytosol. Okay, cytosol only glycolysis takes place, and once glycolysis takes place here, once pyruvic acid is formed, the pyruvic acid has to go on a journey towards mitochondria. Okay, it has to go on a journey to mitochondria where it will be further burnt. So in the mitochondria, mitochondrial matrix, okay, mitochondrial matrix is where the next event of Krebs cycle is going to take place. Okay, and some of them also take place in the inner membrane of mitochondria, like SDH step. Okay, SDH step will be taking place in the inner membrane of mitochondria. So let us say mitochondrial matrix and inner membrane of mitochondria, cristae. So the mitochondrial Krebs cycle take place in mitochondrial matrix. Okay, mitochondrial matrix as well as in the cristae. Okay, mitochondrial matrix and cristae. Then the last event, okay, the last event. So the glycolysis happen in the cytoplasm. Then it goes on a journey. If the pyruvic acid is transported towards mitochondria, wherein it is burnt in the mitochondrial matrix, and further in the inner membrane of mitochondria, cristae, in the oxysome is where electron transport change takes place. So if you just zoom into that cristae, we find in the cristae some equidistantly placed oxysomes, right? They are called oxysomes. These oxysomes are nothing but F0F1 particle. This F0F1 particle is where ATP synthesis takes place. Okay, ATP production takes place. Okay, so that is the last event. Electron transport chain, the last event will happen. It will go on a reverse, goes inside mitochondrial matrix and then reverse journey. Okay, reverse journey. So electron transfer, the last event. Okay, the electron transport chain happen in cristae. It happens in this cristae. Okay, I repeat, glycolysis happen in the cytoplasm. Then the pyruvic acid goes inside the mitochondria, where burning of conversion of pyruvic acid till Krebs cycle, and will be taking place in the mitochondrial matrix, and from the mitochondrial matrix it comes back to the inner membrane of mitochondria, which is cristae, where it is packed with F0F1 particles, wherein ADP will be converted to ATP. So the final climax reaction goes in the last step, which we call it as ATP production happens in the inner membrane of mitochondria, okay, which is called oxysome. Okay, so F0, F1 particles of inner membrane of mitochondria is where electron transport chain happens. So 3 events happen from cytoplasm towards mitochondria. Till mitochondria it happens. So it involves 2 regions. So now let us go for the next step. Because you have to understand 3 events happen in 3 different places. Okay, so we'll go for the next step, that is glucose 6-phosphate to glucose 1,6-bisphosphate. So glucose 6-phosphate to fructose 6-phosphate, an isomer. Okay, fructose 6-phosphate. This one is an isomerase. Okay, so as such, isomers will not involve addition or deletion of molecules or compounds. Okay, here earlier in the kinases we saw phosphate got transferred inside, okay, the reactant, so that the reactant takes up the phosphorus and becomes 6-phosphate, like this one, glucose 6-phosphate. Whereas here there is no change in, just rearrangement of molecules. Okay, rearrangement of molecules takes place, and hence it is catalyzed by phosphohexose isomerase. Phosphohexose isomerase, no involvement of any other molecule. Okay, not even water is involved. Okay, so this is the second step we are talking about, and of course it is a reversible step. We already said the key steps are only 3 irreversible steps, key steps. Okay, there is also a phosphoglycerokinase step also. Okay, so now from fructose 6-phosphate we are going ahead with the next step, fructose 1,6-bisphosphate. So I'll erase this. I hope you have copied this. You have to keep this in mind. Okay, cytosol is where glycolysis takes place, and mitochondrial matrix, the next step, and inner membrane of mitochondria, the next step. Now let us go ahead with the next step. So fructose 6-phosphate will become fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. What is the key enzyme? We said it's a very key enzyme. Okay, yes. The marker is not clear. Marker is not clear. Yes, I'll change the marker. Okay. Fructose 1,6-bisphosphate. Is this clear? Is this visible? Okay. Fructose 1,6-bisphosphate. If it is not visible, you can tell me. Okay. So fructose 1,6-bisphosphate. Again, it is going to be, yeah. Okay. Again, for this reaction, let us see that again there is one more addition of ATP molecule, right? There's one more addition of ATP molecule. Again, addition of phosphorus group. You can see that phosphorus is present only in the 6th position. So there's one more phosphorus in the 1st position. Thereby, it is going to have require one more ATP like this one. Okay. So again, let the ATPs alone, we'll give it in green color. Okay. So ATP is going to come and give the high energy phosphorus donated to fructose 1,6-phosphate to become fructose 1,6-bisphosphate. And this particular enzyme is a very, very, very important enzyme. What is the enzyme? Phosphofructokinase. Phosphofructokinase. The enzyme which is catalyzing this reaction is phosphofructokinase. Phosphofructokinase. I repeat, the first step is glucose to glucose 6-phosphate. By in the presence of ATP to ADP, it give you the enzyme catalyzing it can be hexokinase or glucokinase, specifically glucokinase. Any other hexoses we can call non-specific. We can call it as hexokinase. Okay, then it is converted to glucose 6-phosphate. Glucose 6-phosphate is converted to fructose 6-phosphate, right? Fructose 6-phosphate, and fructose 6-phosphate in the presence of ATP is converted to fructose 1,6-bisphosphate. Okay, fructose 1,6-bisphosphate. Now we are going ahead with the next step. So we have done with the 3 steps, and in the 3 steps, 2 of them are irreversible. I repeat, we only saw 3 steps. In the 3 steps, 2 of them are irreversible. Okay, so again we are going to invest to reap the benefit. First investment, then you can reap the benefit. What is the investment made now? 2 ATP molecules. We are investing it to burn the glucose itself. You have to invest to burn the glucose itself. So, your investment is 1 ATP here, another investment is 1 more ATP here. So, we will be reaping the benefit slowly. Okay. So, periodically, stepwise, we will be reaping the benefit. So, as of now, 2 ATPs are added. Okay, or invested now. Fructose 1,6-bisphosphate is going to get converted to sorry. Okay. So, from here we are going, third step we are in. Now, fructose 1,6-bisphosphate, 1,6-bisphosphate is going to get converted to, yeah. So, this is, we said that it is again an irreversible reaction. Now, fourth step onwards. Fructose 1,6-bisphosphate can be cleaved into, it's a triose, right? Okay. It's a 6 carbon sugar. This 6 carbon sugar can be broken down into 2 3 carbon sugars. So, what are the 2 3 carbon sugars? The 2 3 carbon sugars are, yes, what are the 2 3 carbon sugars? Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay. So, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate and another one is DHAP, which we call it as, again, one is a aldehyde, another one is a ketone, right? One is an aldehyde, another one is a ketone. So, they both are interconvertible. Okay, interconvertible. What is the enzyme which can break one 6 carbon into 2 3 carbons? What is the enzyme which can break one bigger 6 carbon into 2 smaller 3 carbon sugars? What is it called? Which can break one bigger molecule into 2 smaller molecules? Yes. Again, which can break one bigger molecule into 2 smaller molecules. What is the enzyme here, catalyzing here? Aldolase, right? Aldolase. Aldolase breaks the 6 carbon sugar into C3 and C3, but one is an aldehyde, another one is a ketone. Okay, one is an aldehyde, another one is a ketone. And the enzyme catalyzing this interconvertible step is phosphotriose isomerase. Okay, here we said phosphohexoisomerase, but at this step we are going to call it as phosphotriose isomerase. Okay, phosphotriose isomerase, wherein the dihydroxyacetone phosphate has to be returned to glyceraldehyde 3-phosphate for the subsequent reactions to go on. So, both are C3 carbon sugars, and keep in mind, hereafter every step will go in 2 rounds. Okay, so hereafter whatever steps we are going to see, it will be in 2 rounds because it is C3 carbon sugars, 2 C3 carbon. Okay, so 2 C3 sugars. Therefore, hereafter it will go in 2 rounds. Okay, so all the steps from 4th till 10th step will go on for 2 rounds. Okay, anyhow, the DHAP, dihydroxyacetone phosphate, has to be converted to glyceraldehyde 3-phosphate for the reaction to proceed. Okay, for the reaction to proceed. What will be the next reaction? A key reaction. Another very important reaction. What is a very important reaction? What is a very important reaction? Because it is going to involve dehydrogenase. Tell me what class does this particular enzyme that we are going to talk about belong to? Shall we start, have the enzyme? Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Mostly it will start with the reactant. Okay, look at this. Phosphoglucoisomerase. Then here it is phosphofructokinase. Okay, phosphofructokinase. Mostly it will either involve reactant or product. So, hence glyceraldehyde 3-phosphate dehydrogenase is the enzyme which catalyzes this particular reaction. And this particular reaction is very important because there is going to be someone is going to come and receive an important atom from here. Yes, what kind of reaction is a dehydrogenase reaction? Which class does it belong to? Which class does it belong to? Dehydrogenase reaction, which class does it belong to? This enzyme belong to, out of the 6 classes, which class? Yes, I want the instant answers. KG school, I hope the voice is okay now, right? Yes, go ahead with the answer. Which class will this particular enzyme belong to out of the 6? Out of the 6 classes, which class does this particular enzyme belong to? Can I have the answer? Are you all there? Are you all there? Yes, fantastic answer. The answer is none other than oxidoreductase. Comrade School, pat yourself. Wonderful. Okay, wonderful. That was the right answer, of course. Okay, so this belongs to class 1 oxidoreductase. No. MRS, I think you are confused. MRS, it is not hydrolase. Comrade School was the one who gave an instant answer that they are right in giving the right answer. Okay, it can't be a hydrolase because water is not involved. It is just picking up of the hydrogen atoms. So hydrogen atoms are donated and then somebody is going to come and pick up mostly the hydrogens will be taken up by a coenzyme. So answer me this question also. If I am talking about a coenzyme, coenzymes are, listen, answer me this question also. Coenzymes, is it seen there? Okay, coenzymes can be a metal, coenzymes can be a metal, can be a vitamin, can be a mineral, can it be water? What is the answer? What is a coenzyme? Just recall again, we need the knowledge of enzymes here, right? That is why we are roping in the knowledge about. Yes, fantastic answer. Okay, so I got an answer, instant answer, that I think that was Global School who answered that the coenzymes are vitamins. So what is going to be involved here? You are right in answering that coenzymes are vitamins, cofactors are metals. Okay, cofactors are metals, cofactors and prosthetic groups are metals, whereas coenzymes are vitamins and they are loosely bound. Cofactors and prosthetic groups are tightly bound to the enzyme to make it holoenzyme. Okay, yes, very good. You are able to recall all that we have seen some time back about biomolecules, right? Yes. So, who is going to receive the hydrogen atoms? You said dehydrogenase, which means hydrogen atom is pushed out. So, who is going to come and receive? Who is going to come and receive the hydrogens? Because just now we saw a coenzyme and there's a correlation between that and this one. Yes. Who will be the receiver of hydrogen atoms? When a hydrogen is pushed out, somebody should come and receive it. Who is going to receive it? It should be NAD. Okay, NAD to NADH2. Okay, NAD comes in and takes away the hydrogens from this group and it becomes NADH plus H plus H plus. Got it? So it becomes NADH plus H plus. So this we are going to save this NAD. Okay, because we are going to save this NAD for future. We will see that. Okay, later how to burn this NAD, we will see it in the end of the session. Okay, so now glyceraldehyde 3-phosphate. I repeat, glyceraldehyde 3-phosphate. Once because of dehydrogenation, it removes its hydrogen group and finally it is going to become 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate or 1,3-bisphosphoglyceric acid. In the ionized form, we will call it as glycerate. So 1,3-bisphosphoglyceric acid is going to become next 3-phosphoglyceric acid. In that case, there is one high energy phosphorus present in the first group. It is going to be taken away. First time we are going to reap the benefit. You said we invested 2 ATPs. First time we are going to get the profit. Okay, what will be the first profit? In the form of ATP. So this 3-phosphoglyceric acid will be converted to 3-phosphoglyceric acid. 1,3-bisphosphoglyceric acid will be converted to 3-phosphoglyceric acid because the first ATP present in the first group is going to be taken out and ADP becomes ATP. First time we are going to reap the benefit. Here we burnt it. Okay, we burnt it. Here also we burnt it. But first time we are making it. Okay, first time something is, we get the profit in the form of ATP, not just 1, 2 rounds. Remember what we said? Hereafter it will be 2 rounds. So 1 into 2, 2 ATPs. Okay, in this juncture we are going to have 2 ATP and the enzyme which is going to catalyze can always be, whether there is a trans-phosphorus group to or from, it is going to be kinases involved. So what will be the enzyme? Phosphoglycerokinase. Phosphoglycerokinase. Yes, till this copy down so that check what you have done with your notebook and see that it is going on sequentially till here. Hereafter only few more steps to go. Okay, see that it is matching your what you have written in your pen and paper. Okay, cross check if everything goes well. Yes, have you all copied? Shall we go to the next step? So 3-phosphoglyceric acid. So here we are in 3-phosphoglyceric acid. We shall now proceed to the next step. Okay. So how many steps have gone? Only few more steps to go. From 2-phosphoglyceric acid we have to start. Okay. So we can go in the other side of the board itself. From 3-phosphoglyceric acid we are going to go ahead with the positional change. Again an isomer. Okay. What kind of isomer this time? What kind of isomer this time? Functional isomer. There it was aldo-keto isomerism. But here just positional changes. From third position it is going to be shifted to second position. So 3-phosphoglyceric acid will be converted to 2-phosphoglyceric acid. 2-phosphoglyceric acid. And as I said, it is an isomerism, but not aldo-keto, amido-imido kind of isomerism. It is shift in position. From the third position, it is going to be shifted to second position. So this kind of isomerism requires an enzyme. What could be the enzyme? Any guess what could be the enzyme which is going to catalyze this particular step? It is mutase, phosphoglycero. This step is catalyzed by mutase enzyme, phosphoglyceromutase. Are you all with me? Okay, so where are we now? We started here and we have went up to 2-phosphoglycerate. So meanwhile we have reaped the first benefit in the form of ATP. Then 3-phosphoglycerate will be converted to 2-phosphoglyceric acid, and from 2-phosphoglyceric acid this is going to get converted to 2-phosphoglyceric acid will be converted to, in the reaction where it requires enolase, water, it is going to convert it to a high energy phosphoenol pyruvate. And it's indeed a very high energy bond, and the enzyme which is going to catalyze is enolase. Okay, enolase. And water is involved here. Okay, water is involved. So finally we get a very high energy bond and a product with a very high energy bond, that is phosphoenol pyruvate. Okay, enol bond is high energy. So this high energy is going to be donated to ADP to become ATP. So again the last reaction, wherever ADP is involved, it has to be catalyzed by a kinase. So this reaction is going to be by a irreversible step. Again this reaction is all the other steps are going to be reversible steps except 3, as I said. Rate limiting steps are 3 in number. Okay, hexokinase, glucokinase is the first step, then phosphofructokinase step, and the last step pyruvate kinase step. Okay, so this phosphoenol pyruvate is going to get converted to irreversible step by the enzyme pyruvate kinase, wherein ATP is involved, and finally we get pyruvic acid. CH3CO COH. Okay, so we get a pyruvic acid, and this again requires, this is by mutase enzyme. Okay, don't get confused here. Whereas PEP to pyruvic acid is catalyzed by pyruvate kinase. Pyruvate kinase. So we are seeing the fourth kinase now. Okay, so this is the fourth kinase. First kinase here, second kinase here, then third kinase here, then this is the fourth kinase we are talking about. And again here, production of ATP. So 2 investments, then 4 molecules of ATP will be benefited, because you said 2 rounds from here on. Thereby ADP becomes ATP. Okay, here itself I will mark ADP becomes ATP. Okay, so shall we see the net output? Let us see the net output. I repeat, glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, and fructose 6-phosphate will be converted to fructose 1,6-bisphosphate. And these enzymes, 2 enzymes are key enzymes. The first one and the PFK, you make it as PFK, easy to remember. Okay, phosphofructokinase, PFK. Okay, first is HK, second one is PFK, then 1,6-bisphosphate will be cleaved, as it is a 6-carbon sugar with 2 phosphorus. We are going to cleave them into, chop them into 2 equal halves. So glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and this has to be converted to glyceraldehyde 3-phosphate for the reaction to proceed further. So this glyceraldehyde 3-phosphate is going to get converted to 1,3-bisphosphoglyceric acid. This 1,3-bisphosphoglyceric acid is going to donate its phosphorus present in the first carbon to ADP to become ATP. So we get a product as 3-phosphoglyceric acid. And 3-phosphoglyceric acid, just positional isomer, it is going to get converted to 2-phosphoglyceric acid by the enzyme mutase, phosphoglyceromutase. And then water is involved where it converts the phosphoglyceric acid into PEP, phosphoenol pyruvate, and the enol pyruvate is going to get converted to pyruvic acid, and here pyruvate kinase plays a key role in reaping the benefit in the form of ATP. So the net output out of glycolysis, you just take a bank, okay, take a piggy bank, okay, and drop whatever we have. So we are going to have a piggy bank and then drop. So how many ATPs? One here, utilized, right? Utilized. Okay, so 2 here, okay, 2 here, okay. So write it as 2 here, okay, 1A. Then here, 1A, 2B. So we are going to have this 1 and 1 is utilized. Here it is manufactured. It is produced here. It is produced here. Okay, produced here. Thereby 2 rounds, because we said 2 rounds. So 2 plus 2, 4. 4 minus 1 minus 1. 4 minus 2, 2. So finally what is output? We have gained 2 ATP. Is that all that we have gained? No. We have also gained NADH, and again 2 rounds. So this should be 2 NADH. Now out of glycolysis you have saved 2 ATPs and 2 NADH plus H plus. This is the glycolysis savings box. Okay. So out of glycolysis savings box, is it not visible? Okay, glycolysis savings box, we have 2 ATPs and 2 NADH plus H plus. Okay, so this is what we have achieved through glycolysis. Now let us proceed to this pyruvic acid is going to go on a journey towards mitochondria for further burning. Okay, so now let us burn. So far glucose is burnt from 6 carbon state to 3 carbon state. Meanwhile, there is a benefit of 2 ATPs and 2 NADH have been saved in the glycolysis box. With that, we will go ahead with the next one, Krebs cycle. Yes. So with this, we are proceeding to the next step, which is called Krebs cycle. Before that, pyruvate to acetyl CoA step is also there. Okay, so let me erase this and then we will go ahead with, I hope you are doing this same in your notebook as well. Okay, so you can even verify with your notebook as well. So hope that we have done 10 steps so far. Okay, so in the 10 steps, 2 we have got benefit, 2 we have invested, 4 we have got it as benefit. So 4 minus 2, 2 ATPs, we have kept it in the savings bank. Then 2 NADH here in the glyceraldehyde 3-phosphate dehydrogenase step. This enzyme has a coenzyme called niacin or nicotinamide. Okay, nicotinamide adenine dinucleotide. So this niacin is a vitamin B. Okay, so it is a coenzyme of this glyceraldehyde 3-phosphate. So we have even that is the one which received the hydrogens. So NADH plus H plus is the savings that we have done. Now let us proceed with the next step. The pyruvic acid is going to be taken up for the further steps. This pyruvic acid has very many fate. Okay, it has very many fate, that is the pyruvic acid, okay, has many fate. It can get converted to lactic acid upon the enzyme lactate dehydrogenase. Okay, lactate dehydrogenase enzyme can come and convert the pyruvic acid to lactic acid. This happens in muscles during exercise. Okay, this happens in the muscle, and that is the reason for muscular cramps. Okay, in the muscle during exercise, heavy exercise, this leads to the production of lactic acid. So the pyruvic acid will be diverted to make lactic acid in muscles. But otherwise, when aerobic respiration is, again, this is what? Anaerobic respiration, right? Anaerobic respiration. Okay, so aerobic respiration, it has to go through Krebs cycle. Okay, it will proceed through to Krebs cycle using the intermediate acetyl-CoA. Okay, it will use the intermediate acetyl-CoA to proceed to Krebs cycle. Okay, to proceed to Krebs cycle. Then another fate of pyruvic acid is it can also be fermented. Okay, it can also be fermented to alcoholic fermentation. It can proceed even to alcoholic fermentation. So it has 3 options. Okay, so in an anaerobic mode, it prefers lactic acid formation, and in an aerobic mode, it goes to Krebs cycle through acetyl-CoA formation, or if not, it can even proceed to alcoholic fermentation in beverages, alcoholic beverages during wine making, beer making, and other fermented products wherein the pyruvic acid will be converted to the alcohols. Okay, so fermentation, we call that as fermentation. So now we are going to focus on this one, the center one. We are going to focus on this one, how it goes into pyruvic acid, is going into Krebs cycle and extract the maximum energy. Okay, so let us now proceed with this cycle, Krebs cycle. So the first step, the pyruvic acid will be converted to, so CH3 we said COS-CoA, COH, right? So here pyruvic acid is going to get converted to C3 carbon is going to get converted to C2 carbon. So if C3 carbon is going to get converted to C2 carbon, one molecule of carbon dioxide should go out. So this molecule of carbon dioxide will be liberated. Okay, so carbon dioxide goes out. Then what else? Carbon dioxide should be pushed out. Then after all, what you are going to get? Acetic acid is what we are going to get, CH3COOH. Okay, acetic acid is what we will get, but it is not going to be acetic acid, but instead it will become a CoA is going to get added, a coenzyme A. Okay, CoA is nothing but coenzyme A. Okay, coenzyme A is directly can be added. Okay, directly can be added, where it is going to become, instead of acetic acid, it is going to become acetyl-CoA. Meanwhile, there is also a dehydrogenase step. So you have to, it is almost like a decarboxylase. It is carbon dioxide is removed, CoA is added. Meanwhile, there is one more thing, which is NAD goes out. Okay, NAD comes in and then picks up the hydrogens from pyruvic acid and it becomes NADH plus H plus. So the enzyme which is going to catalyze this is a multi-enzyme complex. Unlike a simple enzyme, it's a multi-enzyme complex which is present in the quaternary structure of protein. We saw the 4 structures of protein, isn't it? Primary structure, secondary structure, alpha helix and beta pleated sheet, then tertiary structure, quaternary structure. Mostly the quaternary structures will involve more than one subunit. So this pyruvate dehydrogenase enzyme is a multi-enzyme complex which exists in quaternary structure. Okay, so the enzyme which is catalyzing is pyruvate dehydrogenase. Though decarboxylation also takes place. Okay, though decarboxylation also takes place, the enzyme is named in such a way, it pulls out the hydrogen and then it becomes acetyl-CoA. So it is not going to be CH3COOH, instead it will be CH3CO. Anyhow, one oxygen goes out, so it will be only CH3COH. So it is CH3COS-CoA. Okay, CoAsh. Okay, sulfhydryl group. Okay, so CH3COS-CoA is the acetyl-CoA. So it is a so what we have done is from the C6 carbon, it has been broken to C3 carbon and then the C3 carbon has become now C2 carbon. Okay, C2 carbon. But something is different is going to happen. This C2 carbon is going to go in a cycle. Okay, it is going to go in a cyclic reaction. Okay, cyclic reaction. Okay, which involves oxaloacetic acid, which is a C4 carbon. So C4 carbon and C2 carbon is going to merge to form C6 carbon. And this C6 carbon will liberate the carbon dioxide twice, thereby already one carbon dioxide went out, one more carbon dioxide here, one more carbon dioxide here. Ultimately, what will you say? What will be the key reaction? Key reaction is going to be C6H12O6. We are going to burn it to CO2 plus H2O. Isn't it? How many times CO2? 6. plus X ATP, that is what we are going to find out, plus heat. Okay, X ATP, that is what we are going to find out, how many molecules of ATP are liberated by burning C6H12O6. Okay, so this carbon dioxide, 2 rounds, no? So if you are going to take 6 CO2, it is 2 rounds. Hereafter it will be 2 rounds. Already we said right from the fourth step onwards, it is going to happen in twice. Glyceraldehyde 3-phosphate, from that step onwards, every reaction is going to happen twice, 2 rounds. So in that case, 2 carbon dioxide is going to get liberated here, then 2 more carbon dioxide is going to get liberated. Finally, 1, 2, 3 into 2, 6 carbon dioxide is burnt. Okay, is going to be totally burnt to 6 CO2. What we have to do is we have to only account for this ATPs now. Okay, already we have done 2 ATPs and 2 NADH. Okay, so with that, we'll now proceed to the Krebs cycle. Watch this reaction, so that with that we'll proceed to the next step, which is Krebs cycle. Okay, Krebs cycle. Shall we see the Krebs cycle? So for Krebs cycle, we need a yellow. We have to erase the board and start from the beginning so that we understand the Krebs cycle. Okay, have you all copied down? Is your pen and paper says the same? Is it going to be the same like how you see here? Yes, isn't it? So now let us go for this one. Okay, shall we proceed to the next step, Krebs cycle? Is it done? Is it copied? Is it the same like what you have done in your notebook? Shall we proceed? So from C6 we have burned to C3, from C3 we have now burned to C2, and this C2 we are going to liberate it in the form of CO2 and another CO2. Okay, so CO2 and CO2 again. Shall we proceed to Krebs cycle with this? Okay, now we are going to erase the board and start from the Krebs cycle from acetyl-CoA onwards. From here we will start. So otherwise the board may not be enough. So we will start straight away from acetyl-CoA and go for the cyclic 10-step reaction and then back to oxaloacetic acid, because I said oxaloacetic acid comes in and oxaloacetic acid will be recycled back. Okay, will be recycled back. Shall we go ahead? Yes. Now we will have another bank for Krebs cycle also. We have got one savings bank, one more savings bank for Krebs cycle. So let us start from acetyl-CoA onwards. Acetyl-CoA. Oxaloacetic acid. So this is C4, this is C2, right? C4 plus C2, you are going to get, what is the enzyme which is going to catalyze this? Oxaloacetic acid plus acetyl-CoA, C4 plus C2, we are going to get citric acid, which is going to be a C6 carbon, isn't it? Citric acid. Finally, we get a citric acid. Okay, then citric acid, we are going to have an intermediate, cis-aconitic acid. Okay, cis-aconitic acid by the enzyme aconitase. It's an intermediate, may or may not be included. Cis-aconitic acid. Through an intermediate, finally we are going to have a isocitric acid. Isocitric acid. Okay, so this all are going to be C6 carbons, right? So the enzyme which is going to catalyze this step is citrate synthase and aconitase here. Aconitase, citrate synthase. Citrate, you have to understand, it came from pyruvic acid. Okay, it came from pyruvic acid. Citrate synthase enzyme. Okay, so aconitase, and then we get isocitric acid, right? Isocitric acid is now going to proceed. Shall we proceed on? By the enzyme aconitase, again it is C6. Now we are going to have C5 carbon. First time we are going to liberate carbon dioxide out. Already one is gone between pyruvic acid and acetyl-CoA. Another carbon dioxide is going to get out from this isocitric acid. So the C6 is going to become C5. Okay, shall we make it as C5? So what will be the C5 product? This is a C6 product. C6 product, isocitric acid, and this C6 product is going to become C5 product, alpha ketoglutaric acid. Alpha ketoglutaric acid, glutaric acid, or alpha Kg. Alpha Kg. Okay, and the enzyme which is going to get catalyzed, we said carbon dioxide is liberated out, right? Carbon dioxide is liberated out. Then, meanwhile, there is going to be in the Krebs cycle more than ATP formation. What is happening is more than ATP formation. It is the getting an NADH is the primary role of Krebs cycle. You have to understand there are 3 types of energy formation. Okay, what are the 3 types of energy formation is oxidative phosphorylation, then photophosphorylation, substrate level phosphorylation. Okay, three steps. So what we are looking at here is oxidative phosphorylation, which involves mitochondria. Okay, for photophosphorylation, which happens in plants, require energy in the form of photons. So photophosphorylation takes place in chloroplast. Oxidative phosphorylation takes place in mitochondria. And substrate level phosphorylation. Okay, so substrate level phosphorylation takes place in the cytoplasm. Okay, cytoplasm. Remember, we saw phosphoglycerokinase. In the phosphoglycerokinase, there is a immediate formation of ATP there. Okay, ATP was formed there. So, and here we are going to make GTP here. Okay, GTP will be formed in one of the steps. We call that step as substrate level phosphorylation, which takes place in the cytoplasm. Okay, cytoplasm of the cell. Okay, so mostly phosphorylation will happen in mitochondria or chloroplast. Very rarely it may happen in the cytoplasm, but most of the energy we are able to reap it from mitochondria and chloroplast, very little energy from cytoplasm, which we call it as substrate level phosphorylation. Let us now proceed with this alpha Kg. So in the meanwhile, we said there is an involvement of NAD, right? So the enzyme which is going to catalyze here is ICDH, isocitrate dehydrogenase. Okay, isocitrate dehydrogenase. Whenever we say dehydrogenase, somebody is going to come and niacin is going to come, nicotinamide is going to come and catch the hydrogens and take it with itself. So NAD to NADH plus. NAD plus comes in and then it becomes NADH plus H plus. Okay, NAD comes in and it becomes NADH plus H plus. Okay, you got it? So this is alpha ketoglutaric acid. So this alpha ketoglutaric acid, now it is going to proceed to the next step. It has become now C5 carbon. From here it has to become C4 carbon. Okay, what will be the C4 carbon? Succinyl CoA. So from here, succinyl CoA, we are going to have succinyl CoA as the product. Succinyl CoA. So from C5, we are going to liberate again CO2. Okay, again CO2 is liberated out to make it as succinyl CoA. So again, where will the CoA come? CoA comes in, CoA goes out. So in this step, CoA comes in, and in the next step, CoA will get out. Okay, CoA goes out, and it becomes succinic acid, right? Succinic acid. So let us see this step. So this step, so from here, so 4 plus 2, 6 carbon, which is citric acid, by the enzyme citrate synthase, and this citric acid goes on forming an intermediate cis-aconitic acid in the presence of an enzyme aconitase, so that it becomes an isocitrate or isocitric acid. So this isocitric acid, it liberates the first carbon dioxide out in the Krebs cycle. Actually speaking, it is the second carbon dioxide. Already one is gone here. Okay, already one is gone here. So this is the second carbon dioxide which is going to go out. Okay, so second carbon dioxide which is going to go out here, and meanwhile, when isocitrate becomes alpha Kg, there is also hydrogen liberation. So that hydrogen is going to be captured by NAD, so that it becomes NADH reducing equivalence. We call it as reducing equivalence. Reducing equivalence can be only NAD or FAD. Right? So here it is going to be NAD. NAD is going to come and receive the hydrogens, and it becomes NADH plus H plus. Okay, NADH plus H plus. Now it is from C5 carbon, C6 carbon, it has become C5 carbon. And from the C5 carbon, it has become C4 carbon. Okay, C4 carbon, which is succinyl CoA when the CoA gets added. So again, alpha Kg, this is ICDH we said. Again, this is going to be a alpha Kg DH, which is alpha ketoglutaric acid dehydrogenase. The enzyme which is going to catalyze is again a dehydrogenase. Are you all working out in your notebooks? Okay, alpha Kg DH or DHase, whichever you prefer. Okay, So DHase, you can take it as dehydrogenase. Okay. So alpha Kg dehydrogenase. This alpha Kg dehydrogenase, whenever a dehydrogenase is involved, we are aware that NAD is going to come inside. So NAD is going to come inside. Okay, NAD is going to come inside, and then NAD is going to become NADH plus H plus. Isn't it? NADH plus H plus. And here we are getting succinyl-CoA out of it, right? Succinyl-CoA is going to become succinic acid. So CoA comes in, CoA goes out. Then this succinyl-CoA synthetase. Okay, succinyl-CoA synthetase. The next enzyme is going to be succinyl-CoA synthetase. Succinyl-CoA synthetase. Why there is so much of calm after storm? Okay, we had storm here, isn't it? How about, yes, are you all taking down MRS and global? Okay, there's almost silence from your side. Are you all working out all this? Okay, so succinyl-CoA synthetase. Is there any doubt till here? Okay, so no response from your side. Are you all following this? Are you all working out in your notebooks? I said you start with your pen and paper, isn't it? Yes, you are working out in your paper itself. Very good, well done. Okay, so the succinyl-CoA becomes succinic acid. So that requires succinyl-CoA synthetase. Meanwhile, something happens here. What is it which happens here? Yes, shall I just proceed like that? Shall I just proceed like that? Succinic acid too? With the help of a next enzyme, succinate dehydrogenase. Am I missing anything? Am I missing anything in this step? Succinate dehydrogenase. Okay, succinate dehydrogenase, it is going to become, so succinyl-CoA to succinic acid, CoA comes in, CoA goes out. Now it has become C4 carbon. This is also a C4 carbon. Now all the carbon dioxide went out. Okay, now succinyl-CoA to succinic acid by the enzyme succinyl-CoA synthetase. Is there any other thing happens here? Any other thing happens here in this step wherein succinyl-CoA synthetase is involved? Is there anything happens here? Okay, let me proceed, then you have to tell me. Then succinic acid, again succinate dehydrogenase. Succinate dehydrogenase, this time the receiver is not going to be, usually whenever we say a dehydrogenase, it will be the NAD who will come and take up the hydrogen. But this time it is not going to be the NAD which is going to come and take up the hydrogens. Instead, it will be the FAD, flavins. They are also vitamin B. Okay, most of the coenzymes belong to vitamin B complex. Niacin, flavin, riboflavin. Flavin is nothing but riboflavin. Okay, riboflavin, niacin or nicotinamide. Then thiamine, okay, B1. Okay, thiamine pyrophosphate also is a cofactor. Okay, then of amino acid metabolism. Thiamine, coenzyme A. Okay, so all these coenzyme Q, coenzyme A, they all are belonging to the vitamin B complex. They don't belong to the other one. Okay, we have fat soluble vitamins and water soluble vitamins. Fat soluble vitamins are vitamin A, vitamin D, vitamin E, vitamin K. But water soluble vitamins are WBC, easy to remember, WBC, which means water soluble vitamins are B and C. Okay, so B, in the B, most of the B will be responsible for, in the catalyzing reactions in which they take part in the dehydrogenase steps. So here we call them as reducing equivalents. Okay, reducing equivalents, because they carry not only a proton and an electron. And this, as they carry both proton and electron, that is very, very vital for conservation of energy in the form of ATP. So the protons will be shuffled outside the cell, and the electrons will be transferred inside, so that both protons and electrons. So one reducing equivalent is not just proton is involved. You should not assume that it is only involving hydrogen atoms. Okay, so you have to keep in mind one reducing equivalent involves not only proton, also an electron. So an electron plus proton is what we call it as a reducing equivalent. So here the reducing equivalent here, it is not going to be nicotinamide or niacin, it is going to be FAD, isn't it? FAD. So FAD is going to come in. First time you are using FAD, right? FAD, flavin, then FADH2, or you can even write it as flavor protein, FPH2, whichever you prefer. Okay, you can also write it as FP to FPH2, H plus, FPH plus H plus, like that. Okay, flavoprotein or riboflavin, flavin adenine dinucleotide. FAD to FADH2, because we are using FAD for the first time here. That is why I made a change, different color. Okay, FADH plus H plus. So here I said succinyl-CoA synthetase involves substrate level phosphorylation. That's what I said. Something is missing here. Just remind me, I said. So here is where substrate level phosphorylation takes place. Here what happens is there is going to be a GTP formation here. Succinyl-CoA synthetase. When the succinyl-CoA becomes succinic acid, here GTP is formed. GDP comes in and then it forms GTP. Guanine. Okay, all the while we will be talking about adenine trinucleotide, adenine ATP, not GTP. So we have two purines, right? Adenines and guanines are purines. We are aware of that, isn't it? Purines. So always most of the energy currency is in the form of ATP, but very rarely GTP also is involved. So here GTP is formed. Okay, here it is GTP which is formed at this juncture, where this GTP straight away we can even stop with GTP, or we can convert the GTP into ATP. Keep in mind, GTP is formed in this step, plus ADP will form GDP plus ATP. You understand? This phosphorus is donated to ADP so that it becomes ATP, and GDP is again, we get a GDP again. So we can consider this particular step as we are getting a profit of 2 ATP. Actually speaking, it is 2 GTP, but we can also consider it as 2 ATP, because as I said, GTP plus ADP can give you GDP plus ATP. So we can even consider straight away as we are receiving 2 ATPs in this step. Okay. So what is the name for this? Krebs cycle, then tricarboxylic acid cycle, then citric acid cycle, isn't it? All of them mean the same. Citric acid cycle, all of them mean the same. Yes, let us proceed to the further steps. Where are we now? Succinic acid. Okay, succinic acid dehydrogenase converts succinic acid to fumaric acid. Fumaric acid, right? Then fumaric acid will get converted to malic acid. Malate. Okay, malate. Malic acid or malate. And malate again here just involves water. It involves only water. Water is thrown out. And by the enzyme fumarase. The enzyme which is going to catalyze here is fumarase. Okay, fumarase. Now we are getting malate, and malate finally recycles the oxaloacetic acid back. So OAA, oxaloacetic acid, is recycled back from malic acid. We started with oxaloacetic acid, and now look at that. The acetyl-CoA which came in is burnt out as carbon dioxide once, carbon dioxide twice. Then that's all. Only two carbons only came. It all became carbon dioxide and carbon dioxide. Meanwhile, there is a lot of NADH which we are getting it from this Krebs cycle. So Krebs cycle, the idea of Krebs cycle is to make as many reducing equivalents as possible, because you can see that ATP molecules produced from this Krebs cycle is very less. Only GTP is produced. Okay, so it is mostly in the form of reducing equivalents. Okay, where are we now? Malate. So the malate will be converted to oxaloacetic acid. So the oxaloacetic acid, and here what do we call it as? What will be the enzyme which is going to catalyze this? Malate to oxaloacetate. The enzyme is again going to be MDH. Malate dehydrogenase. Okay, MDH, malate dehydrogenase. So I repeat, we have been seeing a lot of dehydrogenase now in this cycle. We only saw one in glycolysis, glyceraldehyde 3-phosphate. But here we are looking at many dehydrogenases. Okay, so again, NAD is going to come in, and it is going to capture the hydrogens, and then it is going to become NADH plus H plus. NADH plus H plus in this step, and oxaloacetic acid is recycled back by this step, malate dehydrogenase. So how many dehydrogenases are there? Okay, there are very many dehydrogenases in this step. Okay, now we are going to look at it. Let us see the overall picture of Krebs cycle, and then we will see, we will go for the energy chart. Okay, so pyruvic acid is a 3-carbon sugar, gave 1 carbon in the form of carbon dioxide, and it became acetyl-CoA when the CoA got added. So it's a decarboxylation step, and also we get NAD in this step also we reaped NAD, isn't it? Because it is NAD, pyruvate dehydrogenase step. So we also got NADH plus H plus in this step. Then afterwards this 2-carbon sugar went for a cyclic reaction wherein it got mixed with, it combined with oxaloacetate, and the oxaloacetate has to be recycled back in the end of the steps. So oxaloacetic acid is a 4-carbon sugar. It combined with C2 carbon, which is acetyl-CoA, and it becomes citric acid. So CoA came in, CoA got out. Okay. So as a citric acid. So here CoA, so acetyl-CoA, and again here the CoA is lost. Like here in this step we said CoA came in, CoA got out. Same way CoA got removed, and then finally we get citric acid, and this citric acid is a 6-carbon sugar. This 6-carbon sugar became another 6-carbon sugar, isocitric acid. Okay, isocitric acid. Meanwhile there is an intermediate which is formed, cis-aconitic acid. Then this isocitric acid will be converted to C5 carbon alpha Kg. Okay, C5 carbon alpha Kg. This alpha Kg is very, very vital because this we find alpha Kg. From alpha Kg, glutamic acid will be produced, okay, in amino acid metabolism. So this alpha Kg is an intermediate of many other metabolism, including amino acid metabolism, where from alpha Kg we can also get glutamic acid, okay, glutamic acid and amino acid for protein synthesis. Glutamic acid can be received from alpha Kg also. Okay, so let us see for aminotransferase reaction, transaminase reaction. So in transaminase reaction, this alpha Kg can also be converted to glutamic acid. Okay, come back here. So the isocitric acid in this sugar metabolism, isocitric acid is converted to alpha Kg, and meanwhile one carbon dioxide is lost. Already one. This is the second one. Okay, so second one, carbon dioxide is lost. Thereby C6 will become C5 carbon alpha Kg. And the enzyme, whenever we say dehydrogenase, there is going to be an NAD involvement. NAD is the one who is going to come and receive the hydrogen atoms in the reducing equivalents. So the reducing equivalent is NAD here by the enzyme isocitrate dehydrogenase. Then we get alpha Kg, and from alpha Kg, succinyl-CoA. So when we say succinyl-CoA, C5 carbon is going to become C4 carbon. When C5 carbon becomes C4 carbon, one carbon dioxide is again lost. So back to C4. We started with C4, ended with C4. C4 came here itself as C4. Okay, succinic acid, yeah, succinyl-CoA. So C4 itself. So again the enzyme is going to be alpha Kg dehydrogenase. Whenever dehydrogenase is involved, we already aware there is going to be an involvement of reducing equivalent NAD. So NAD is going to come in, pick up the hydrogen atoms, and then it becomes NADH plus H plus. Then CoA got added, it became succinyl-CoA. Okay, so succinyl-CoA will be converted to succinic acid. So CoA, whenever CoA is added, it promotes metabolism. Okay, so you'll be wondering why should CoA be added? Why should CoA be removed? In the first step also we said CoA added, CoA subsequently got removed. Same here we say CoA got added, so CoA got removed. So addition of coenzyme A promotes metabolism, promotes burning of sugars or burning of any compound. So it promotes metabolism. That is why CoA gets added and then CoA gets removed. So okay, same way the CoA got added here and it became succinyl-CoA. Now hereafter it is going to be only C4 carbon. Okay, C4 carbon alone. Okay, the C4 carbon again, CoA, after removing CoA, it becomes succinic acid. Meanwhile, you have to keep in mind there is a formation of ATP. Not directly, indirect formation of ATP takes place because GTP is formed here by the enzyme succinyl-CoA synthetase. Okay, so GTP is formed and the GTP can also be added with, as we saw in the first step in the previous slide, we saw it as GTP plus ADP can also form ATP plus GDP, right? So succinyl-CoA synthetase, it became succinic acid, right? Succinic acid by the enzyme succinate dehydrogenase, it became, again the reducing equivalent is not going to be NAD, this time it is going to be riboflavin. Okay, flavin. So flavin is going to pick up the hydrogens and it is going to become FADH2. Then fumaric acid. Then fumaric acid, water is removed and finally by the enzyme fumarase, we get a 4-carbon sugar malate. And this malate, here there is no involvement of reducing equivalence, only water is involved. Okay, no energy is involved here in this step in the conversion of fumaric acid to malate. From the malate you get oxaloacetic acid. Okay, so recycled back, oxaloacetic acid came in, now it is recycled back. Okay, so by the enzyme malate dehydrogenase, and here also NAD is going to be involved. NAD is the reducing equivalent, and it is going to pick up the hydrogens and electrons from the from malic acid, and it is going to convert that malic acid into oxaloacetic acid. So we are going to have malate dehydrogenase finally, which is getting converted wherein the oxaloacetic acid is recycled back. Now let us go for a chart, energy chart. So already we have savings. We have saved 2 ATPs, 2 NADH. Shall we save it for now? For Krebs cycle, are you all ready to save for Krebs cycle? Now take a bank. We are going to put in the savings. What are the savings that we can drop in this savings bank? First one. 1 NAD. Look at it carefully. 1 NAD in the first step. Okay, 1 NAD in the first step. Then second NAD. Okay. Second NAD. Then third NAD. Okay, third NAD. Fourth NAD. Check yourself. See if you are getting 4 NADs in your notebook. Check if you are getting 4 NADs in your notebook. Are we getting 4 NADs? Right from pyruvic acid. Pyruvic acid 1 NAD. Then here 1 NAD. Here we have NADH plus H plus. Here also 1 NADH plus H plus. Check if you are also getting the same. Are you getting the same? NADH plus H plus? Yes. So we are getting 4 NAD. So we are dropping. So is it just 4? Is it just 4 or 4 into 2? Is it just 4? Because we said from the glyceraldehyde step itself, everything is going to go in 2 rounds. So it will be in 2 rounds. So 4 into 2, 8 NADH plus H plus. Am I right? NADH plus H plus. So 4 into 2, because 2 rounds. So 4 into 2, 8 NADH plus H plus. Okay, fine. Now what is the next one? Okay, 8 NADH plus H plus. Now is that alone that we receive from here? No. We also receive from succinate dehydrogenase 1 FAD. So 1 into 2, 2. Here. This step. Okay, so 1 into 2, 2 FADH plus H plus. Okay, 2 FADH plus H plus, right? Then what is this one here? We saw GTP, right? Either you can take it as GTP or, as I said, you can even take it as 2 ATPs, 2 rounds. So we will consider that as 1 into 2. Okay, 1 into 2, either GTP or ATP. Okay, this is what we have received out of Krebs cycle. Are you all getting the same? Are you all getting the same kind of energy reducing equivalence we received? Because as I said, this cycle does not produce much ATP, rather it produces only the reducing equivalence, whereas glycolysis produced 2 ATPs and 2 NADH, and in this reaction we got more of NADH plus H plus. Okay, is that clear till here? I hope this is clear now. Shall we proceed to the next level? What is the next level? We are going inside the inner membrane of mitochondria cristae. Shall we proceed into the inner membrane of mitochondria cristae? Now we are going on a journey, where from the cytoplasm we went to the mitochondrial matrix, right? So mitochondrial matrix, this cytoplasm of mitochondria is called mitochondrial matrix. This is where most of the Krebs cycle reaction took place. Some of them took place in the cristae also. this one SDH for example. Okay, so now from the matrix we are going on a journey back again to the inner membrane cristae, because this is where ATP is going to be produced. Okay, ATP is going to be produced in the cristae, inner membrane of mitochondria. Now we are going on a journey, reverse journey. We went up to mitochondrial matrix, then we are coming back to the inner membrane of mitochondria and also the intermembrane space. Okay, this is intermembrane space. Okay, this region between outer membrane and cristae, we call that as intermembrane space. Okay, in the intermembrane space also, our next reaction is going to involve two space. One is inner membrane and another one is mitochondrial matrix, and one more is intermembrane space. Okay, now let us proceed all this, whatever we have saved here, we are going to take it and bring it to the next cristae. Okay, so these boxes we will carry. There are two boxes we have saved, one on the left, one on the right we have saved. We are going to take these two boxes and take it to the mitochondria. Okay, shall we take it to the mitochondria? Yes, we are going to take it to the mitochondria. I am going to erase this so that we will proceed on to inner membrane of mitochondria. Then we will answer those questions which we have posed. Yes. Okay. Now we are going to have two boxes. We have two boxes now. Okay. I'll put both the boxes on the one side. This is the pyruvic acid and Krebs cycle, pyruvic acid to Krebs cycle box. Another box we had is glycolysis box, in which we saved 2 ATP and 2 NADH plus H plus. Okay. So this is our saving so far. Now we are going to take it to the inner membrane of mitochondria. Shall we see the inner membrane of mitochondria? Equidistant. They are placed in equidistant. What is this? What is this we are talking about? This is where ADP is going to get converted to ATP. So I said the ADP gets converted to ATP only in the inner membrane of mitochondria. Okay, so here till here pyruvic acid came, and the pyruvic acid was here, and then it got converted to acetyl CoA. From the acetyl CoA it gave out carbon dioxides, and finally it is coming here and taking these NADs and FADs, and it is going to convert this NADs and FADs into ATP. Okay, there's going to be a shift in which the reducing equivalents are going to get converted to ATP. Okay, are you all there? Yes, what is this stalk and knob called? What is this called? What is the stalk and knob called? In the inner membrane we have the stalk, and I'm zooming this, okay, one of them. Okay, it should be throughout, not that it is only on one side. Okay, so don't assume it is only present on one side. It is present throughout the inner membrane of mitochondria. Okay, inner membrane of mitochondria, it is present in an equidistant way. Okay, yes, what is this? What is this? What do we call this as? What is the stalk and the knob? What is it called? Because all this we are going to take it there only, in the inner membrane of mitochondria, cristae. Yes, what is this called? F0, F1 particle. Okay, what is this called? F0, F1 particle of inner membrane of mitochondria cristae. This is where ATP production takes place. This is where ATP synthesis takes place. Okay, so what is it? Is there another name for this? Can you tell another name for this F0, F1 particle? I have given blanks there. Okay. Maybe I can fill up some so that you can, it ends with SOM. Yes, now tell me, the F0, F1 particle can also be called as this. It ends with SOME. You have to just prefix it, prefix the 3 word and tell me the answer. The inner membrane of mitochondria is filled with F0, F1 particle, wherein, yes, fantastic answer there. Very good answer there. Oxysomes. Okay, oxysomes is the answer. Fantastic. Oxysome is the answer. Oxysome, Kamraj, very good. Okay, so oxysome is the answer for F0, F1 particles, another name. Okay, another name for F0, F1 particle present in the inner membrane of mitochondria is oxysome. Fantastic answer there. Okay, now we are taking all these saved energies and we are going inside the mitochondria. Shall we go inside the mitochondria? I'm going to zoom in this mitochondria. Okay, this inner membrane. Cristae is going to be zoom in. Shall we zoom in? Yes, we are going to zoom the cristae. Mitochondrial inner membrane. So what is the kind of ribosome present in mitochondria? I only give 2 options because you are talking about mitochondria. We have to know about certain things, isn't it? So, inner membrane of mitochondria has ribosomes, isn't it? Its own ribosomes. What is this ribosome? Is it 2 options only, 7ts or 8ts? The ribosomes of inner membrane of mitochondria is 7ts or 8ts. Tell me. Yes, am I getting an answer? There are ribosomes here, right? So, the ribosomes present here, I am asking, is it 7ts or 8ts? Because this is present inside a eukaryotic cell. Keep in mind, we are talking about a mitochondria which is present in a eukaryotic cell, not a prokaryotic cell. Because we know prokaryotes will not have mitochondria. Prokaryotes will not have organelles. Okay, bacteria do not have organelles. Only eukaryotes will have organelles. I am asking you an option. The ribosomes are present in mitochondria. Is the ribosome 7ts type or 8ts type? Yes, go ahead answering me that question before we could draw this picture. No, it takes time to draw this picture. By the time you can answer me that question. Only 2 options are given to you. Is it only 7ts or 8ts? That's all. The ribosomes of mitochondria is 7ts or 8ts? Yes, go ahead answering that question. Somebody's answered there. Yes, fantastic answer. Good. 7ts. Okay. So 7ts is the answer. My answer is 7ts is the answer. Though it is inside eukaryote, every other eukaryote, the ribosomes present in eukaryotes are 8ts, whereas the ribosomes present inside mitochondria is 7ts. Okay, good. Okay. Yeah, good. Even 7ts. I think Kamraj has answered already. Okay. So the ribosomes present inside mitochondria is 7ts, but whereas the ribosomes present in all other regions inside eukaryote is 8ts. Okay. Come back here. By the time we will draw this complex 1. Say complex 2 is very, very unique. Let us have another color for it. This is complex 2. Okay. Then we will go for complex 3. Okay. Shall we see what happens in the inner membrane of mitochondria? So this complex 1 is NADH dehydrogenase. Complex 2, complex 1 is NADH dehydrogenase, 2 is succinate dehydrogenase. Succinate dehydrogenase. 3 is cytochrome BC1, and 4 is cytochrome AA3. Cytochrome AA3. Okay, I repeat. 1 is NADH dehydrogenase, 2 is succinate dehydrogenase, and 3, complex 3 is cytochrome BC1, complex 4 is cytochrome AA3, wherein water is formed. Okay, water is formed by taking in oxygen and the protons. Water is formed. It will be reverse in the case of plants. Plants split water. I repeat, the first step for light reaction is photo-splitting of water, isn't it? Water is split to get oxygen. So what do we do here? So it's almost reverse. If you compare photophosphorylation with that of, this is oxidative phosphorylation, isn't it? So what happens in plants is, if you compare plants, okay, for oxidative phosphorylation and photophosphorylation, you will find the first step itself is photo-splitting of water in plants. Okay, this is making water. Okay, water formation is in the last step. Okay, it's the last step for oxidative phosphorylation in animals. It's the last step. Okay, whereas in the photophosphorylation of plants, photophosphorylation happening in the mesophyll cells of plants, first step itself is splitting of water, photo-splitting of water. Okay, photo-splitting of water. Then, what are the differences? Photo-splitting of water. What are the differences? This is the first step, this is the last step in animals, oxidative phosphorylation. This is the first step in plants. Okay, in plants wherein photo-split, water is split. Then what is happening here in oxidative phosphorylation? We evolved 3 into 2, 6 molecules of carbon dioxide. Carbon dioxide is evolved. What do plants do? They purify the air. We spoil the air, they purify the air. But they too, during the night time, they too will produce carbon dioxide. Same like animals, they too produce carbon dioxide. During the daytime, they harvest the energy. During the night time, for the transport and for all the enzymes to function, they will burn the sugar that they have stored. So during the night time, they will evolve carbon dioxide, same like us, whereas during the daytime, they purify the air. Oxygen is evolved. Okay, oxygen is evolved, but we produce carbon dioxide and they produce oxygen during the daytime. Then first step we said, last step it is water is involved. Water is made, but water split. You can see, we make water, they break water. Okay, we make water, water is made in the last step, right? Water is produced in the last step, whereas here in the first itself they break the water to get energy. Okay, the photo-splitting of water takes place. Then what are the other differences? Here we have ubiquinones. Ubiquinones will be present in oxidative phosphorylation as electron transfer, electron carrier, whereas here plastoquinones are involved. Okay, plastoquinones are involved. Ubiquinone, there plastoquinone. Then what will be the other differences? What are the other differences? 4. Then cytochrome cytochrome BC1 and cytochrome AA3 are involved, whereas in plants, cytochrome B6F is involved. Cytochrome B6F is involved. Okay, so we are seeing 4 differences about oxidative phosphorylation and photophosphorylation, right? So here water is made only in the last step, but in the case of plants, water is broke in the very first step itself. And here we produce carbon dioxide. They too do the same. Okay, plants also do the same during the dark reaction. Okay, during the night time. Okay, during the daytime, they evolve oxygen. They purify the air. Then here they have electron carriers like ubiquinones, whereas plastoquinones are electron carriers in photophosphorylation. Then cytochrome BC1 and cytochrome AA3 are the electron carriers of oxidative phosphorylation, whereas in the case of photophosphorylation, cytochrome B6F is the electron carrier. So likewise, there are differences between oxidative phosphorylation and photophosphorylation. So we are going to follow this up with the C3 cycle and C4 cycle and CAM cycle, light reaction and dark reaction. Okay, so what we have seen is what is happening in all animals, including plants during the daytime. But what we have not seen is C3, which is going to happen light reaction, and the C3 is going to happen in plants, which we will see it in the next class. Okay, now we'll proceed it with electron transport chain. Made a comparison between oxidative phosphorylation and photophosphorylation. Now let us proceed with those two boxes, what we can do about it. Okay. Okay, so look at this. Now, the complex one, as I said, is NADH dehydrogenase. Maybe a schematic representation will help you in understanding this. Substrate comes in with hydrogen and donates the hydrogen. Substrate goes out. What is the substrate? It can be alpha Kg, or it can be malate, or it can be, what are the substrates? Substrates could be pyruvic acid, pyruvate, first step, no, pyruvic acid, or it could be isocitrate, or it could be alpha Kg. It's not succinate dehydrogenase. Then because it is going to involve only in the second step, so alpha Kg, then malate. It can be anything. Anything is going to come, donate the hydrogen to and then to whom? NAD. Oxidize, reduce. And the substrate is going to go back. Then NADH plus H plus. NADH plus H plus. Again oxidized, reduced. Oxidized, reduced, you keep on drawing. Then there's a lot of iron sulfur center here. Iron sulfur center. Oxidized, reduced. Then what will be the next one? Carefully observe this. We are just, whatever diagram we have drawn here, we are making a schematic form, easily understood form. Here we are writing it parallelly. Okay, so iron sulfur centers are present in the complex 1. We are talking about complex 1. So iron sulfur center rich. Then we have this here. There are also ubiquinones. We just now said there are also ubiquinones, but whereas in the plant system there are plastoquinones. Now the second complex, we are going to come to the second complex. So here what happens is, after the substrate donates the hydrogen to NAD to become NADH plus H plus, there is going to be shuttling of hydrogen from inside to outside. Proton is going to go from I'll make it a little wider for you to understand, okay? The protons which are present here is going to come out from inside to outside. From the inner membrane, protons are going to move out to the intermembrane space. To the intermembrane space, the protons are going to get moved out. So there will be accumulation of protons in this intermembrane space. Okay, when there is metabolism, when the glucose is oxidized, okay, when the glucose oxidation takes place, what happens is proton accumulation happen between the outer membrane and inner membrane of mitochondria in the intermembrane space. Okay, in the intermembrane space, accumulation of protons take place. Okay, so now, shall we proceed on? Now there is going to be proton which is present here, which is taken from here, is going to be shuttled outside. When one proton is released out, there is a chance of conversion of ADP to ATP because of the proton gradient, as proposed by Mitchell's chemiosmotic hypothesis. Who proposed this? Mitchell's chemiosmotic hypothesis says that, chemiosmotic hypothesis by Mitchell says that the proton gradient produced, can you see a proton gradient? A proton gradient produced is enough to convert ADP to ATP. So this proton gradient is pivotal for the conversion of ADP to ATP. Okay, so now you got it. So now that iron sulfur centers, now coming to the complex 2. The complex 2 is FAD. Okay, so flavoprotein is going to come because flavoprotein as such, FAD comes in and then it goes as FADH2 from the substrate, succinate dehydrogenase. Okay, from the substrate intermediate, it comes from succinate, you have to keep in mind. Like how we said it comes from pyruvate, isocitrate, alpha Kg malate. So this comes from succinate. I think the board is not visible. So this comes from succinate. It becomes FADH2 plus H plus. And this reaction is unique. You have to understand there is no proton shuffling from complex 2 So it loses 1 ATP. Listen again. So the succinate merges, succinate to FAD merges only in the complex 2, and there is no proton shuffling from this complex 2, so it misses 1 ATP. Keep in mind, I'll explain you in the end. Okay, come back here. Now we have a question also to be answered, right? Come back here. So now that FAD, now we come to complex, there is cytochrome BC1. Okay, cytochrome B, oxidized, reduced, cytochrome B, then, okay, I think black is better, right? Then cytochrome C1, oxidized, reduced, then ubiquinones, oxidized, reduced, then we have last one, cytochrome AA3. I'll put it together because lack of space. Cytochrome AA3, oxidized, reduced. Meanwhile, oxygen plus protons will form water. Oxygens plus protons will form water. Okay, can you see here? So meanwhile, you also have to keep in mind there is going to be shuffling of protons in BC1. BC1, there is going to be proton will be pushed from inside to outside. Already we said there is one proton is going to be pushed out from the inner membrane towards the intermembrane space once here. Then once more here, okay, between BC1, okay, after BC1, there is going to be a proton shuffling. Then also towards the end, from cytochrome AA3, there is going to be proton shuffling. Listen, so proton is going to get shuffled. I think it becomes clumsy there. Okay, it is going to get shuffled from the first complex, protons. Second complex, no. Okay, no shuffling from here, no. Then complex 3, proton shuffling is going to take place. Okay, because succinate joined only in the second step, it loses the first proton shuffling, thereby it loses 1 ATP. So there is going to be a proton shuffling from the third complex, and also from the fourth complex there is going to be proton shuffling. Can you see here? Is it understood? Forget this part. Okay, so it is understood that cytochrome BC1, cytochrome ubiquinone, cytochrome AA3. So this is represented as this. I think I can erase this. Okay, erase the bottom so that you can straight away understand it from the picture that we have drawn here. Okay, from the picture we have drawn here. In complex 1, there is shuffling of protons. Complex 2, no. Complex 3, there is going to be shuffling of protons, and complex 4, there is going to be shuffling of protons. Okay, I'll erase the bottom because it looks shabby here. Let us erase this. I hope you have understood what I meant. Okay, so now what happens is the protons are moved out, the electrons roam around here itself. Okay, so the electrons roam around till it reaches the end. Okay, so the protons are shuffled. So for NAD, you can see that all the protons which are moved out, okay, all the protons which are moved out towards the intermembrane space will return it back by the enzyme in ATP synthase, F0, F1 ATPase. ATP synthase, it will funnel all the ATP protons which are accumulated in the intermembrane space back to the mitochondrial matrix inner membrane, wherein ADP will be converted to ATP. Are you clear? So the protons which are shuffled to the intermembrane space will return back to the inner membrane. Meanwhile, when there is a gradient, proton gradient created, that proton gradient will be conducive enough to convert ADP to ATP. That is what Mitchell's chemiosmotic hypothesis say. Okay, so now let us see. So as a rule of thumb, one NAD will give rise to, one NADH plus H plus will give rise to how many ATP? 1 ATP here, 1 ATP here, 1 ATP here. Okay, so 3 ATPs. Keep in mind, one NADH plus H plus will give rise to 3 ATPs. Okay, 3 ATPs. As succinate joins only in complex 2, it loses the ATP it has to receive from the complex 1. So, it will only receive the ATP from complex 3 and complex 4. Therefore, 1 FAD gives rise to 2 ATPs. Therefore, 1 FADH plus H plus gives rise to only 2 ATPs. Are you all clear? Keep in mind, as a rule of thumb, one NAD, how? You have to understand how it is able to give 3 ATP, how this is able to give 2 ATP. So, as a rule of thumb, NAD gives rise to 3 ATP, FAD gives rise to 2 ATPs only, because it loses the ATP from the complex 1 as it joins only from complex 2 onwards. There is shuttling of protons only in the complex 3 and complex 4. So, for 1 proton, 1 ATP. So, finally we get 2 ATPs for FAD and 3 ATPs for NAD. Okay, is that clear? Is that clear for all of you? I hope this is a hectic session, I understand. Okay, so I hope that this part is clear for all of you. So, shall we take those 2 boxes and work out on the energy? Are you all ready? We have only few minutes left. Okay, shall we work out on the energy chart for we have saved in some 2 boxes, right? We have to convert that in the form of energy and try to answer the first question. Compare aerobic and anaerobic respiration and tell how efficient it is. Okay, that was the first question we posed, isn't it? So, I hope now you are clear with the electron transport. What are we looking at? This is nothing but mitochondrial electron transport chain. Mitochondrial electron transport chain, ETC. So, initially we saw glycolysis, second we looked at Krebs cycle, now the last step is mitochondrial electron transport chain. The last one is ETC, mitochondrial electron transport chain. We started with glycolysis, went to Krebs cycle, and before that pyruvic acid to acetyl-CoA step, then we went to the cyclic reaction of Krebs cycle. Then after Krebs cycle, we took all the NADH and brought it to mitochondria inner membrane, where the mitochondrial inner membrane converted that into ATPs that we are going to look at in the last step. So, this step is called mitochondrial electron transport. So, 3 steps: glycolysis, then Krebs cycle and electron transport chain. All together we call it as burning of glucose. Okay, aerobic respiration. Starting from glycolysis till ETC, we call it as aerobic respiration. These 3 steps combined, we call it as aerobic respiration. Now, let us convert these boxes. Okay, let us focus on these boxes. I think that the mitochondria step is understood. I am going to erase all of this and then take those 2 boxes and convert it into the form of ATP, right? Yes. Shall we take this to boxes? Yes. So, glycolysis. In glycolysis, we had 2 ATPs, then 2 NADH. As a rule of thumb, 1 NAD gives 3 ATP. So, 2 NAD gives 2 into 3, which is equal to 2 into 3 ATP, which is equal to 6 ATP. So, 6 plus 2, overall 8 ATPs. So, net profit is 8 ATP molecule from glycolysis box. Okay, from glycolysis box, this is what we have achieved. Let us go for the next box, savings box. We are going to pull out the second box and then take this box. Okay, convert this box. Shall we convert that box? Take that box. Okay, so that is from pyruvic acid till Krebs cycle. Pyruvic acid till Krebs cycle. We are going to pick up that box and we found that there are 8 NADH plus H plus that we have retrieved. Okay, so 8 NAD means 8. 1 NAD gives 3 ATP as we all are aware. So 8 into 3. What is 8 into 3? 24 ATPs. Then 2 FAD. Okay, 2 FAD. So, FAD we know it is only going to give us 2 ATP. So, 2 into 2, 4 ATP. Okay, then already there was GTP produced. Okay, we can even take that into account as 2 ATP. So, 2 ATP. Straight away 2 ATP was already there. ATP or GTP. Okay, so altogether how much? Okay, all together. From this Krebs cycle alone, 30 ATP. Then already 8 ATP. So 30 plus 8. So from glycolysis. So aerobic respiration means from glycolysis till ETC. From glycolysis till ETC, electron transport chain. In that case, this is 8 and this is 30. So finally, output is 38 ATP. So what is our final output? We received 38 ATP molecules. Is that clear? So 8 ATP molecules from glycolysis, then 30 ATP molecules from Krebs cycle, from pyruvic acid to Krebs cycle. So 30 plus 8, 38 ATP molecules is what we have received. Okay, 38 molecules together 38 molecules. And what was our first question? When we began our session, there was a question which I posed. I said we will answer it in the end. When you compare aerobic respiration with that of anaerobic respiration, okay, in terms of energy, how efficient is it? Is it 2x? Is it 18x? Okay, I think I can recall the question. What the question was asked is compare aerobic with that of anaerobic respiration. If you compare aerobic with that of anaerobic respiration, the number of ATP generated is efficiency is 2x, 18x, 26x, then 32x or something. Okay, 38x. I don't recall anything. Okay, anyhow. So let us consider it as like this. Okay, what will be the answer is? What will be the answer? 18x or even say 19x. Okay, 19x because 38 ATPs, right? So the number of ATPs produced in anaerobic respiration is, this is aerobic respiration. What you are looking at is aerobic respiration, 38 molecules. The number of ATPs generated in anaerobic respiration is 2 ATPs. So it is how many times efficient? 19 times or 18 times? Even if you don't consider the GTP, okay, if you don't consider the GTP, then it becomes 36 ATP. Okay, then it becomes, excluding GTP, it becomes 36 ATP. So in that case, the answer should be 18 times efficient or 19 times efficient. Okay, 19 times means it will come exactly to 38 ATP. Okay, so sometimes they also don't consider the 2 GTP. In that case, 36 ATP. Okay, if the GTP is not considered, okay, then it will be 36 ATP. So 19x or 18x. They won't give 18x and 19x together. So the answer should be either 18x because the number of ATPs generated in anaerobic respiration, wherein pyruvic acid gets converted to lactic acid is, here we get 2 ATPs. So overall only 2 ATPs are received in glycolysis. Okay, 2 ATPs only are received because pyruvic acid becomes lactic acid. Okay, so in that case only 2 ATPs out of glycolysis. 4 minus 2 is equal to 2, right? So in the glycolysis box we had 4 minus 2 is equal to 2. So 2 ATPs only from glycolysis. So the final output itself in anaerobic respiration is only 2 ATPs, whereas in aerobic respiration it is 38 or 36 ATP. So the efficiency should be in terms of how many times is 18 times or 19 times. So it is, they will never give 18 and 19 together. So it should be 18 times, or if they look for 36 ATP. If you consider 36 ATPs, it is 18 times. If you consider 38 ATPs, then it is 19 times efficient. Have we answered now? Okay, I think I got an answer as 32 or 38x. No, it is not that efficient. Okay, it is not that efficient. So this is wrong. So the answer should be 18x or 19x. 19x or 18x is the answer for this. And we are able to get energy from glucose in stepwise release of ATP. It is not in one go. In one go, glucose will not be burnt out to give 38 ATPs in one go. It goes periodically in a coordinated manner. You have seen how many steps, isn't it? Already 10 steps in glycolysis, 10 steps in Krebs cycle, and here also we have seen 4-5 steps. So together 20 to 30 steps, 30 steps only will convert glucose to ATP, 38 molecules of ATP, okay, in a periodic manner. So it cannot give in one go, glucose will not give out 38 ATPs. In stepwise, small stepwise reaction, it releases energy, and the maximum energy that we get out of one molecule of glucose is 38 ATP molecules. I hope you have understood, and moreover, as it is there in your syllabus also, this will be very much useful to you. Okay, but only thing is you have to work out with your pen and paper. Once more, if you just take your pen and paper and work out from the glucose all by yourself, do it yourself, from glucose till electron transport chain, till we get metabolic water. Okay, till we get metabolic water. If you go till this step, then you can be very, very thorough with from glucose till water, metabolic water formation in oxidative phosphorylation. In our next class, we will see what happens in a plant. Okay, photophosphorylation. How does photophosphorylation take place in plants? And also we will see the light and dark reaction, C3 cycle, C4 cycle, and crassulation acid metabolism in our next session. Thank you.